Okay, so we'll get going with Ben's talk. I know some of you have just joined for Ben's talk, and some people have been here for the NeuroSig meeting before, so we've just had updates on the Division of Neuropsychology, on the different neuropsychology courses around the country, and I think Ben and I are probably part of the evidence base for the benefits of having cohorts, because Ben and I sat beside each other for most of our Glasgow training course and doing the knowledge component and doing the clinical component together. So it's my pleasure to introduce my study buddy, Ben, who is going to talk to us about rare mitochondrial disorders today and talk about the mechanisms, pathology and neuropsychological consequences.
Thanks very much, Colleen. I'm assuming everyone can hear me, but just add a comment in the chat function if people can't or if it's not clear. So thank you very much for kind of inviting me. It's obviously quite novel doing this via Zoom. I've made the assumption that a lot of people don't know a lot about mitochondrial disorders, even if you have seen someone who has a mitochondrial disease. So I've kind of just tried to cover everything sort of broad church, but also with the context of I've actually only been in the post for 7 months, so I'm very much a novice myself. But we'll go through it. There is a pause kind of in the middle for me to check the chat if there's particular questions, rather than me kind of skipping in between the two. So yeah, we'll make a start anyway. Okay, so a little bit about the service. I work at the Newcastle NHS Highly Specialised Service for Rare Mitochondrial Disease. I work there part-time as a highly specialised clinical psychologist based with the neuropsychology department at the Royal Victoria Infirmary Hospital. I work as part of that highly specialised multidisciplinary team, so it's made up of neurologists, nurse specialists, speech and language therapists and physiotherapists. And one day a week I also work with the national charity, the Lilly Foundation, who I'll mention in a little bit more detail as we go along. And I obviously had to get a picture in of the North East and just show it off a little bit because people are much further south than I am right now. Okay, so what I'm going to kind of plan to talk about today is firstly, kind of just a definition of terms, just because genetics it was a very new area for me and it might be a new area for people. I'm going to talk a little bit about mitochondria, so their origin and their cellular function and how that fits in with the bigger picture. I'm going to talk a little bit about mitochondrial dysfunction and pathology, so particularly kind of neuropathology and about how the mutations and symptoms present. I'm going to talk about cognitive consequences, so looking at some of the research that's been complete around some of the kind of cognitive impact of this condition. And finally, the psychological and systemic factors, which there's not a lot of evidence for, but I thought, seeing as I'm in this post, I might as well offer some initial thoughts anyway. Okay. So, some important terms. So starting off with genome. So the genome is all of the genetic material of an organism. DNA, I'm not going to try and pronounce or say it. I've tried my best, but I trip up every time. Chromosome, so a DNA molecule of which there are 23 pairs, the 23rd of which determines sex, so XX and XY, which people will be familiar with. And lastly, gene. So this is a segment of DNA which is needed to contribute to a function rather than its location. So exactly what it does for the person, the individual, and how that then interacts with the environment. So I don't expect you to remember that, but I will refer to these terms as we go along. Okay, so initially DNA. So all genes are composed of DNA. Each rung or ladder consists of 2 paired chemicals called bases. And there are 4 types: adenine, thymine, cytosine, and guanine, or A, T, C, and G, and you'll see why those are relevant a little bit later on. Each base is symbolized by the first letter of its name for obvious reasons. And certain base pairs always pair together, so A and T and G and C always pair together. The sequences of base pairs form coded messages, a bit like kind of spelling a word. Okay, so genes. A gene is a sequence or a string of bases. Combinations of A, T, C, and G, as I've already mentioned. And the unique combinations determine the gene's function, so much as letters join together to form words. And I'm going to use kind of spelling as a bit of an analogy throughout this, because that's a good way of understanding mitochondrial disease. So each person has thousands of genes, so billions of base pairs of DNA. And these determine individual characteristics or genetic traits. So I hope again that's familiar to people. So what are mitochondria? I'm going to have to desperately try not to say the powerhouse of the cell, because I'm sure there's some kind of a joke around there about that, but I'm going to end up saying it. So mitochondria are rod-shaped organelles that are the power generators of the cell. And they convert oxygen and nutrients that we take in into adenosine triphosphate, or ATP for short. So it's this process of energy release that's very important to our function, but also why mitochondria are there. So the power stations, they're the power stations, sorry, in virtually every human cell. So people refer to them as almost the battery of the cell. And they create energy from fuel and oxygen. And this energy is needed to fuel every biochemical reaction that occurs in the human body. The consequences of depletion of DNA, mitochondrial DNA, is that mitochondrial energy is impaired, which can lead to dysfunction of several organ systems, which I'm going to talk a little bit more about. So everything that the body does, from staying warm to moving and thinking, to sweating in the heat, to taking a drink, whatever it might be, it comes from proteins, carbohydrates, and fats. And we are unable to use these directly, but instead convert them into a form of energy that cells can use, called ATP, as I've already mentioned. And again, you can think of this like a battery, and this converts chemical energy into a different form, in this case electrical, that is easier to use. And for our bodies, the conversion from food to energy to ATP, that's what happens in the mitochondria. If your mitochondria are not working properly, then you are less able to convert food into ATP. And for cells that require a lot of ATP, for example, your muscles, your brain, this is a problem, and that can become weaker and get tired faster. So the number of mitochondria in a cell can vary. So in red blood cells, there are absolutely no mitochondria, none at all. However, in liver cells, there are more than 2,000. And although most of a cell's DNA is contained in the cell nucleus, the mitochondrion has its own independent genome that shows substantial similarity to bacterial genomes, which I didn't know before I got into this kind of area, and it's very relevant to how we understand mitochondrial disease. So why? Why does it have a different genome? So mitochondria and chloroplasts likely evolved from these things called engulfed prokaryotes that once lived as independent organisms. And at some point, a eukaryotic cell engulfed an aerobic prokaryote, which then formed an endosymbiotic relationship with the host eukaryote, gradually developing into a mitochondrion. I'm glad I got through those sentences in one piece. So what does that mean then? So ultimately, without mitochondrion, without these kind of weird bacteria type things that joined us billions of years ago, we simply couldn't and wouldn't exist. Simply, we wouldn't have had the energy to develop into human beings. So here is the human genome in all of its glory. I'm not going to begin to try and explain or understand that, but these are all of the different A, T, C, and Gs that make up us and our characteristics. The mitochondrial genome is much smaller, but it's completely different in every way, shape, and form. So the DNA in our mitochondria will ultimately be very different to the DNA that you take from the rest of our tissue or bodies. And again, I'm going to break this down a little bit into how that relates then to mitochondrial disease. So what is mitochondrial disease? So mitochondrial disease is a chronic, long-term genetic, often inherited disorders that occur when the mitochondria fail to produce the energy that's needed for the body to function properly. They can present at birth, but can also occur at any age. So the service that I work in is a kind of birth to death service. We see everybody. 1 in 5,000 individuals has a genetic mitochondrial disease, and each year about 1,000 to 4,000 children in the US are born with a mitochondrial disease. With a number of type of symptoms and organ systems involved, they're often mistaken for other more common diseases, which I'll refer to when I talk about the psychological consequences. So they're very difficult to diagnose, and they often present very, very differently. So when we talk about the two different types of DNA, we talk about nuclear DNA, which is the human genome, and we talk about mitochondrial DNA. So mtDNA is that mitochondrial one that I was talking about. So mitochondrial DNA disorders are said to affect a minimum of 1 in 10,000 adults in the UK, although other reports suggest the burden of disease may be much more common. And again, this is some of the challenges of diagnosis, which I'll talk about as we go on. So what causes mitochondrial disease? So under normal circumstances, a child inherits genes in pairs, one gene from the mother and one from the father. A child with a mitochondrial disease does not receive a normal pair of genes from the parents. The gene is mutated, so it's become defective. So that, again, thinking of the spelling analogy, it's that the code that's written is incorrect. So learning the way that a mitochondrial disease has been inherited helps predict the chance of passing the disease on to future children. I'm going to break that down a little bit now. So inheritance then. So in autosomal recessive inheritance, the child receives one mutated copy of a gene from each parent. So there's a 25% chance that each child in the family will inherit a mitochondrial disease. In autosomal dominant inheritance, the child receives one mutated copy of a gene from either parent, and there is a 50% chance that each child in the family will inherit a mitochondrial disease. But you also get random mutations. So sometimes genes develop a mutation of their own that is not inherited from a parent. And again, understanding that is a big part of working in the service, about understanding kind of pedigree and how people inherit these conditions. One of the more challenging parts of mitochondrial disease is what I'm moving on to now, which is mitochondrial inheritance. So this is the mitochondrial DNA that's inherited. So in this unique type of inheritance, the mitochondria contain their own DNA, as I've already mentioned. So only mitochondrial disorders caused by mutations in the mitochondrial DNA are exclusively inherited from mothers, exclusively. And if this is the way that a mitochondrial disease was inherited, there is a 100% chance that each child born in the family will inherit that disease. And again, that's going to come on to kind of some of the systemic factors I'll talk about later on. Okay. So what are the symptoms of mitochondrial disease? And I'm going to kind of go through some common ones here. Firstly, heart. So people with mitochondrial disease can present with cardiomyopathy, Wolff-Parkinson-White syndrome, which is the heart kind of beating abnormally fast, and conduction disorder. They can have problems with the gut, so they can get pseudo-obstruction or gastrointestinal dysmotility, so the whole system working very, very slowly. People can get Pearson syndrome in the blood, which is, it's characterized, it's kind of an anemia that also affects the pancreas, so can lead to diabetes, and diabetes is very common in this population. The eye, so optic neuropathy, ophthalmoplegia, which is to do with the muscles of the eyes, and retinopathy. Liver, so hepatopathy, which is disease of the liver. And obviously why I'm here talking to you all, because the brain, it's an organ that uses significant amounts of energy, and therefore the symptoms can include seizures, myoclonus, ataxia, stroke, dementia, and migraine at varying different levels. So it's a very complicated condition. So this is where I go into kind of the heavy territory. This is what I thought would impress people who were interested into neuropsychology. So if it's tedious and boring, I'm sorry, but at least I learned something while I was doing it. So neuropathological mechanisms then. So there are many different approaches to understanding the mechanisms of mitochondrial neurodegeneration. Ultimately, mitochondrial disease is a neurodegenerative condition. It is a life-limiting illness. So understanding those mechanisms is part of the work of the team, and also how we understand the progression as well. So the mitochondrial membrane potential generated by these things called proton pumps, so complexes 1, 3, and 4, is an essential component in the process of energy storage during oxidative phosphorylation. Don't ask me any questions on that bit. Oxidative phosphorylation is the metabolic pathway in which cells use enzymes to oxidize nutrients. So ultimately releasing energy, which produces ATP. So this takes place in the mitochondria, as I've already said. And almost all aerobic organisms carry out this process. And it's so pervasive because it's a highly efficient way of releasing energy. Kind of short story, our body is incredible at maximizing the amount of energy that goes in and how we use it through our mitochondria. Together with the proton gradient, this kind of membrane potential forms the transmembrane potential of hydrogen ions, which is harnessed to make ATP. And I promise this has got a point. It's heavy at the moment, but we are leading into it. So what does that mean? So mutations of the mitochondrial DNA affecting subunits of mitochondrial complexes impair the efficiency of this respiration, so how the energy is being taken up. so basically it's a slow system. It's just not burning the energy that it needs. And maintenance of this potential is essentially what our mitochondria are there to do. So cells lacking this DNA maintain a modest membrane potential despite being able to perform very little of this. So they can do something, but it's much reduced. And this is the important bit. So discharge of mitochondrial membrane potential has a number of consequences for the cell, including apoptosis. And this is a form of programmed cell death or cellular suicide. So it's different from necrosis, in which cells die to injury. It's an orderly process in which cells' contents are packaged in small packets of membrane for garbage collection by immune cells. So it's a very slow process of degeneration. So severe mutation in complex 1 changes the mechanism of maintenance of mitochondrial membrane potential. So again, that energy burning in neurons and astrocytes. And this potential impairs that kind of processing of oxygen and oxidative processing. So enhanced consumption of ATP in response to pathological situations such as seizures, which are common in mitochondrial disease, might be potentially harmful for neurons because they're just not managing the oxidative process. So severely deficient cells demonstrate a combination of impaired complex 1 and high mitochondrial membrane potential. And this combination increases production of reactive oxygen, and excessive generation of free radicals ultimately leaves the neurons vulnerable to damage by oxidative stress. So that was a really heavy, complex way of going through, of just saying the mitochondria, the energy that it's using, too much, too little, is very damaging to neurons and astrocytes in particular. And therefore neurons die more than astrocytes because of oxidative stress. So if you've made it this far, we're going on to something a little bit lighter. Again, it's probably satisfied the scientist in me in explaining some of that anyway. Okay. Right, so common mutations. I thought it would be good to talk about some of the common mutations that we see and some of the signs and symptoms as well. So if we go back to that mitochondrial genome, where all of the arrows are, if you can see my mouse moving, this is the diagnosis. So if you diagnose with M.15555A to G, which would be in this segment, you would present with deafness. That would be the phenotype. As you can see, when I first started in the service, I panicked quite a lot, thinking, how am I going to begin to understand this? 7 months in, I'm getting a bit better, but I'm certainly not an expert. But I'm going to go through some of these diagnoses in turn, just so you can begin to get a sense of how this might affect a person and what you might see in clinic. So the first one is something, the most common one, which is M3243A to G, or MELAS. People might have heard of MELAS in particular. So it stands for mitochondrial myopathy, encephalopathy, lactic acidosis, and stroke-like episodes. And it's the most common variant, with and without MELAS, because not everyone with this condition has MELAS. And it's also related to other conditions and several other mutations, but these are the type of symptoms we would see. Symptoms typically develop during childhood, however they can begin at any age, and it's characterized specifically by recurrent stroke-like episodes in the brain. And these aren't strokes as we would typically expect. These are metabolic episodes because of these kind of free radicals and oxygen depletion. So these are the symptoms. Seizures, headaches, vomiting, appetite loss, muscle weakness, fatigue, vision, hearing loss, dementia, aphasia, and motor skill deterioration. It's a whole kind of multi-system problem, but there are some specific features that are kind of interesting for neuropsychology. And that is focal occipital seizures, and they're the hallmark of MELAS. So visual hallucinations can be really, really common. And they may be associated with transient or permanent visual field defects as well, or other posterior cortical symptoms. The seizures may have focal motor features, or the presence of prominent posterior interictal discharges and photoparoxysmal responses suggest that the posterior occipital parietal regions are significantly involved. So that isn't a case that I've seen with the images at the top, just something that I found online just to show the area of kind of damage, deterioration. So in MRI scans, it shows lesions, particularly in the parieto-occipital region, and these can cross vascular territories and are often transient. They usually spare white matter and show increased signal intensity on T2 weighted images. Okay, so the next common mitochondrial disease, I'm not going to run through all the numbers every time because it's, as you can imagine, it's exhausted, but this one's commonly referred to as MRF for short. And that stands for myoclonus epilepsy associated with red ragged fibres. And this usually occurs during childhood and has an effect on the central nervous system and skeletal muscle. It's characterised specifically by myoclonus, so sudden, brief, jerky movements in the arms, legs, or entire body. So symptoms can include muscle weakness, muscle stiffness, ataxia, seizures, peripheral neuropathy, and eventually dementia. So people do experience neurodegeneration quite rapidly. Again, I'm not going to read the numbers out, but this condition is called LON for short, which stands for Leber hereditary optic neuropathy. And LON is central vision loss. So the vision loss may begin in one eye or simultaneously in both eyes. And people usually present with blindness after diagnosis within the first couple of years. Yeah, so severely impaired within 3 or 4 months. Peripheral vision usually remains unaffected in this condition. And other symptoms include painless blurring or clouding of vision, colour vision loss, and blindness, as I've already mentioned. Another condition that might be common that people hear about is Lee syndrome, particularly in children. And this is a progressive disease that begins in infants and typically results in death within 3 years. In very rare cases, an individual may survive into their early teenage years, and it's characterised by kind of the loss of mental and movement abilities. And there's some of the symptoms there. So again, kind of really kind of difficult and upsetting symptoms for families to manage. Okay. And I think the final one that's common is POLG, or polymerase gamma, but POLG for short. And this is the enzyme responsible for mitochondrial DNA repair and replication. So mutations in this gene can lead to quite a number of different mitochondrial diseases. So that includes Alpers-Huttenlocher syndrome, which is progressive organ dysfunction, ataxia neuropathy spectrum, which, as it says, it's people become increasingly, or children become increasingly more ataxic. I'm not going to read them all because you can read them yourself, but you can hopefully begin to understand in my description how complex this is. And I'm still tripping myself over some of the big words as I go, but hopefully it's helpful in kind of explaining some of the common things that come up. Okay. I think we're getting there. So this is the reality of everything is, it's an incredibly highly heterogeneous condition. Whether that's related to age, phenotype, lifestyle, it's just incredibly different all the time. And a multitude of neuropsychological issues present, in my experience to date, but there do remain a lot of unknowns, so about genetics, prognosis, diagnosis, symptoms and symptom management, and importantly, I guess I think being one of the first people to work in a specialist service, even though people have seen these patients, starting to think about some of the psychological and social consequences, as well as some of the neuropsychological issues. So this is a little break from all that. Brief pause, that's how I described it. I thought if we get through all the science, then we can have a little break and maybe ask some questions. I was very curious just about if anyone had any questions. I can see one pop up from Ingram. And also what experience people had with this population, if any. I know when I spoke to Cleaner about doing this, I was just very curious nationally and having this kind of forum to see who's seen people with mitochondrial disease. So if people have the chat function and want to kind of pop any comments into the whole group or just to me privately, that would be great. So Ingram's seen a few people. And Ingram, you asked what explains the phenotypic variation. Again, I guess it's to do with kind of the location of the mutations, age, lifestyle factors, but also some things that we don't quite know yet, I think, in my experience. Certainly I don't know enough about it to understand how we begin to understand phenotypes. And certainly seeing neurologists say regularly in MDT meetings, we've not seen this before, is interesting because yeah, some things we just haven't seen before. So the phenotypic variation is interesting. So someone's seeing a gentleman with MELAS and a younger onset dementia service, which again is probably quite common. Sorry, I'm reading and you've just got my blank expression. Someone said you're doing a good job and you should write this up. Thanks. I'll try my best if I get some time. Just started working in a metabolic service and I've already had a few new metabolic variation with an interest in neuropsychological profile. How is it diagnosed? So again, we work with a woman with MELAS in an inpatient setting at Putney. So that's really interesting as well. So thanks. I will move on unless people are typing. Flying through now. I regret giving everyone the free rein all at once, maybe.
There's a question from Ingram about working with children and an adult service, Ben.
So that's a really good question, Ingram. So I am very much on the adult neuropsychology spectrum, hence doing kind of the Glasgow practice component at the moment. So it is at the moment an adult bias service, which is a shame, but my post is kind of a development post with the view of developing services very much, I think, to have a paediatric neuropsychology component in it. We have paediatric neuropsychology colleagues in the team who I do refer to, and they do see those patients. Unfortunately, some of our younger population are usually very impaired, and it's much more about the systemic work anyway, so I will see families.
There's a question from Nora about how is it diagnosed, Ben?
Yeah, I'm going to come on to that a little bit later on. There's various different ways it's diagnosed, muscle biopsy being one of the kind of best and most common. But one of the psychological challenges I'm going to talk about later on is the diagnostic odyssey, they call it, because people getting a diagnosis of mitochondrial disease is incredibly difficult. Yeah, and someone said there, Catherine, about remember the trauma of the time pathway to actual diagnosis. Yeah, I think you're right as well, Ingram. Definitely find an appropriate model for neuropsych work into lifespan conditions. Absolutely agree. I think I've sort of got my work cut out over the next couple of years, but we're just really beginning to think about kind of mitochondrial diseases and understand it. And I think that's why coming to something like this and just trying to create a bit of a kind of a national interest in this and trying to connect the dots with people helps us better, I think, create a pathway and services and etc. So with that in mind, it's not getting any cooler, so I'm going to move on. If that's all right with everyone, I'll have some time for questions at the end. So I'm now going to talk about the cognitive and psychological literature. I might fly through the cognitive stuff just because it's all kind of test data and results from studies, but it'll be there for people to read if they'd like to, and I'm happy to send the paper on if people would like that as well. So one of the best papers on cognition in mitochondrial disease is this one by Muir. And they wanted to determine the cognitive profile of patients with M3243A to G and M344A to G. So that's MELAS and MRF, which again, I think are the most common conditions that people might have seen, but also to try and assess disease severity. So they used a prospective case control design. So there's 36 people with MELAS, 13 with MRF, and then they also used match controls, of which there were 32. So they assessed 3 times over 18 months using the WAIS, the WATAR, the DKEFs, and the WIMS. So the results. I am going to fly through this, so sorry for just kind of rushing through. But what they found is patients performed significantly lower on general cognitive functioning compared to the normative group. And there was impairment at 1 standard deviation and 2 standard deviations. They performed significantly worse on all WAIS-4 subtests. And perceptual reasoning performance was significantly better than processing speed. Executive functioning when compared to the norm group. So verbal fluency, patients performed significantly lower with higher than expected rates of impairment at 1 standard deviation. And again, there's some details there about impairment at 2 standard deviations. Again, I think you're getting the headline that they generally are performing on all kind of measures worse than the normative group. And verbal fluency category and verbal fluency switching correct did not differ significantly, indicating normal verbal fluency switching performance when accounting for semantic fluency. So here's another question. Okay. They also did the DKEF's tower test. So on the DKEF's Tower test, patients showed significantly worse scores than the norm on all subtests, but the Tower move accuracy ratio. So again, there's something about being slower here, about having more significant problem-solving issues. There was impairment at 1 standard deviation, but also impairment at 2 standard deviations. So sorry if I am kind of flying through this, but again, I'm happy to send the paper, and I've got a summary at the end of this section. So what about memory when compared to the norm group? So patients scored significantly lower than the normative population. Again, it's a bit of a trend. So patients demonstrated poorer free recall than expected in relation to recognition memory. Again, impairment at 1 standard deviation and impairment at 2 standard deviations. I think what I'm going to do is just fly through this section actually, just because it can be quite repetitive, and I'll go to the summary because I think that's much more interesting. Okay, so summary results. So in terms of predictors of cognition in patients with mitochondrial disease, there was mild to moderate premorbid difficulties. There were significant changes to verbal comprehension, perceptual reasoning, working memory, processing speed. And that was even after correcting for motor speed difficulties. Compared to matched controls, patients performed worse on all domains except memory and executive function. And again, the significant part there is helpful to know. And patients demonstrated significant memory retrieval difficulties. Executive function performance was variable, and impairments may be caused by other difficulties, so difficulties with word generation, decision-making being slower but accurate. So again, just this general slowing down because of the reduced mitochondrial energy consumption. And interestingly, the results of MUA are in contrast to other studies that report clear executive dysfunction. And interest in this study found that it might be better explained by slower decision-making all round. I think that reduced processing speed is probably the most significant factor in this group in my experience. So although immediate and delayed recall were significantly worse than the norm, heightened recognition performance demonstrated difficulties with memory retrieval rather than encoding or storage. So sorry I rushed through the other slides. It would have been helpful to talk through those, but I think I just wanted to kind of get that summary in whilst being mindful of time as well. Okay, so this is kind of the last section, thinking a little bit about the psychological consequences and considerations in mitochondrial disease. And this is, as far as I'm aware, there's very limited research in this area, but we'll present the research that is available. So the first one is around fatigue, very much from a psychological perspective. So the aim of this study was to determine the prevalence of fatigue and perceived functional impact in mitochondrial patients. It was a cross-sectional postural survey of 132 people, and the data was compared with healthy controls and patients with ME or chronic fatigue syndrome matched for age and gender. And the method was they used the fatigue impact scale, the Epworth sleepiness scale, and the HADS. So the results of this study, 62% of patients with mitochondrial disease reported excessive symptomatic fatigue. So fatigue impact scale of over or equal to 40. 32% reported severe functionally limiting fatigue symptoms, comparable to perceived fatigue patients with ME and CFS. So what about quality of life? So again, the aim of this study was to determine the impact of mitochondrial disease on quality of life. So it was 72 patients with MELAS or M32438 to G, and they completed some questionnaires. Those are the questionnaires there. So again, summary of results. Patients reported impaired quality of life. 60% showed severe levels of fatigue, which is consistent with other research. 37% showed clinical relevant mental health problems, which were significantly worse than healthy norms. And these patients reported health outcomes which showed negligible relationships with levels of heteroplasmy. So this is the load of the condition in blood, urine, buccal, or fundamentally how much the disease has progressed, and weak to moderate relationship with clinical manifestation or phenotype. One of the questions that's interesting is about depression, and I hadn't appreciated, I guess, as a psychologist, about mitochondrial DNA, energy, about what impact that might have on depression, per se. And there's a question about the neurochemical impact of mitochondrial disease on mood. There's some emerging evidence in the paediatric population of this. However, the consensus thus far is it's very difficult to establish whether depression in an individual with mitochondrial disease is one of the CNS or a consequence of chronic systemic mitochondrial energy depletion or due to a separate gene defect leading to the expression of the mood disorder. And already I'm hopefully speaking to lots of psychologists who are saying, what about the psychology of all of it as well? It's not just genes and systems and other things. And I think really there's a gap in the research there for us to understand systemic factors in thinking about depression. And kind of lastly, in thinking about this area is neuropsychiatric difficulties. So in an Italian study, in patients with mitochondrial disease, psychiatric conditions were far more common than in the general Italian population, about 60% versus 20 to 25%. And that included major depression, agoraphobia, panic disorder, generalized anxiety disorder, social anxiety, and psychotic syndromes. Psychiatric involvement did not seem to depend on disease progression. Without kind of sharing too much about a clinical case, but in my experience, because there's a lack of awareness about mitochondrial disease and how it presents, particularly with the occipital lobe seizures, people can present with visual hallucinations or have changes in their visual landscape, which I think some psychiatry colleagues are assuming is psychotic in nature without understanding the kind of neuropsychiatric link. And even more so when I've spoken to neuropsychiatric services, in my experience, and it has been limited, people haven't necessarily heard or come across mitochondrial disease. As we've already mentioned, so the process of diagnosis, which I think is really helpful to talk about. So participants who took part in this study saw an average of 8.19 clinicians. The first clinician consulted about symptoms was typically a primary care physician, although 35% of participants initially sought a specialist. 55% of participants received their diagnosis from a neurologist, 18% from a clinical geneticist, and 11.8% from a metabolic disease specialist. Interestingly, over half received one or more non-mitochondrial diagnoses before their final diagnosis. And in their pursuit, 85% received blood tests, 71% received a muscle biopsy, 60% an MRI, 38% urine organic acids. In addition to that, 40% of the participants underwent mitochondrial DNA sequencing, which is no mean feat, and 19% sequencing of nuclear genes. 11% had the whole exome sequence, so the lot. A bit of a fishing exercise, potentially. But you can understand there the process to diagnosis for patients, as I think you pointed out, Catherine, is harrowing, difficult. I don't know much about muscle biopsies, but from what I've heard from people who've had them, they're horrendously uncomfortable and painful, and in itself can be quite traumatic to go through, particularly then when it results in a diagnosis of mitochondrial disease. So this paper concluded that the diagnostic odyssey of patients with mitochondrial disease is complex and burdensome. It features multiple consultations and tests and often conflicting diagnoses. And these reflect disease variety, diagnostic uncertainty, and clinician unfamiliarity. So these are some specific psychological factors that I also picked out that I thought would be helpful just to kind of talk about in my experience to date. There's no evidence yet that I'm aware of. I'm going to try and find some, or at least create some if I can. But this group in many ways is no different to those living with a long-term health condition or a neurodegenerative condition. So they have to manage great uncertainty, physical symptoms, fatigue, pain, loss of function, cognitive changes, as well as a myriad of other factors relevant to having a chronic health difficulty. I guess navigating the benefit system, our current economic climate as well. However, there do seem to be some very specific psychological factors relevant to this population. So in my experience, firstly, it's complex guilt. So when we think about mitochondrial DNA inheritance and it exclusively coming 100% inheritance from mum, unfortunately, I've seen families where mum is affected by the condition, all children are affected, and the husband is unaffected. And unfortunately, in some cases, husbands have seen their entire family gone within a decade. Also kind of women who've passed on this genetic condition unknowingly, living with the guilt of that as children then present with these really complex, distressing health conditions. So that's one area that I think can need some more attention and how we work with it. Relevant to that then is significant systemic loss. So similar to Huntington's disease, the population have seen many parents and siblings lost to the disease, and that rattles through families. And even more so because this is such a new area and there's so much new technology that helps us understand, potentially seeing loved ones die many years ago and we didn't know why, or we didn't understand why. So something like cardiomyopathy, it might have been a heart attack that they died of, but they might have been degenerating prior to that. Body image. So because of ophthalmoplegia and particularly changes to the eyes, people have ptosis, which is drooping of the eyelids. Because the body's not producing enough energy, or it's not converting fuel into kind of extra muscle, I often see people who are very short stature. And obviously myoclonic jerks, which you'll be familiar with, are difficult for people being mistaken for being drunk. And all of these contribute to changes to identity in particular. And lastly, living with a rare disease. So there's a general lack of understanding when accessing local health services, benefit systems, and navigating the health system more generally. A big driver of mine in coming in this post was doing things like this. As much as it made me feel anxious and a bit nervous, etc., it's trying to raise awareness of these issues so that actually if you were to see someone in clinic who's, say, like a cognitive clinic where someone's presenting with new symptoms, your mitochondrial hat might be on and you might think, is this relevant and who can we speak to? So again, about the service. So we provide specialist national provision to adults affected by the condition. So we have people travel from Wales, Scotland, Northern Ireland, and I'm very interested to develop a network of professionals interested in mitochondrial disease to hopefully improve the kind of scope and quality of services, and hopefully eventually complete some research. It always seems to be, particularly when you're a clinician, the thing that's at the back of the pile in terms of what you're doing. So I'm trying to do some more practice-based work. And if you have experience of the condition and would like to discuss a case or make a referral or learn more about mitochondrial disease, then please get in touch. And this was kind of one of my big aims of coming to speak to you all today. Finally, there's currently a storyline happening in Coronation Street about mitochondrial disease, which you may or may not be aware of. The Lilly Foundation are doing some amazing work, as are the Wellcome Trust for Mitochondrial Research, at kind of informing the writers. They've been involved throughout and making sure that this is an accurate portrayal. And because of that, the Lilly Foundation are receiving a number of incoming calls from people who are affected by this condition now that there's a more kind of national profile. But obviously we're in the middle of coronavirus. It's been challenging for all charities across the services that we see. But if you do have a moment to kind of check out the Lilly Foundation, find out the work they're doing and make a donation, that would be incredible. Interestingly, I think Coronation Street gets 14.9 million viewers. And we were just reflecting that if every viewer gave £1, that would be incredible. So if you were kind of interested in supporting, even if it was just because you've enjoyed this kind of me talking about mitochondrial disease, that would be fantastic. So I have very much rattled through that, but thank you. And I appreciate there might be some more questions. My NHS email address is there, and also my Twitter handle if you'd like to follow me on Twitter. I try to be as active as I can. So again, yeah, I hope it wasn't too science heavy. Hope it's been interesting. And if you do have questions, you can either ask now or contact me afterwards. So thank you very much.